Hello students, welcome to EPG Pathshala University Grant Commission. I am Dr. Sohail Akhtar from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard, New Delhi. Today we are going to discuss about a module, Introduction to Osmotic Drug Delivery System, which is the part of Controlled Drug Delivery System. So it is under the paper Novel Drug Delivery System 1. So students, let us see what we are going to learn in this module. This module will discuss or will tell you about the introduction to the osmotic control drug delivery system, what is its advantage, principle behind the development of osmotic control drug delivery system, and what are the important components of such drug delivery system. So before going into the detail, let's have a look into its basic structure, or its basic about the drug delivery system. In the past few decades, the rate of occurrence of various diseases has been increased exponentially. To overcome these health-related problems, uh, researchers, biomedical scientists, and pharmaceutical industry are trying hard to find out a new way or new kind of drug, and not only the new kind of drug, but also their effective mean of delivery for obvious reasons, like achieving three main reasons like safety, quality, and the efficacy for that particular drug. So there are a lot of drug that has been discovered, marketed, and subjected for post-marketing surveillance. Post-marketing surveillance means it is in the clinical trial and it is now in the phase 3 before covering the major area or major population. So when a drug is in the market and being prescribed to patients, then only long-term side effects can be noticed. So that the next challenge for researcher is to lessen this side effect. Okay, that for those drugs which are in market for longer term. The other main hurdle for most of the drug to be a blockbuster molecule are high dose, low water solubility, less bioavailability, and short elimination half-life. So therefore, a smart tailored drug delivery system should release or should have the property to release active pharmaceutical ingredient, that is the drug, in sufficient amount at the same time, also along with the reduction in the side effect to some extent. Among all the route of administration, most widely route of administration that we know is the oral route, because it provides the best compliance to the patient. But the patient encounter with different type of problem related with this particular route, such as the medication is not completely released. Sometimes the drug also causes some serious adverse effects when it reaches into the site of action or site of delivery. Moreover, the frequency and dose is also high, and the minimum effect concentration of drug reaching to the desired site for lesser period of time. Most important things, this drug, that is the oral route, although it is most convenient, however it is not appropriate for any drug which is gastroirritant. One thing. Another thing is that it is also not an appropriate drug delivery system for making or targeting a particular organ, because you are going to lose majority or most fraction of drug into the GIT system itself. The oral bioavailability of a drug can be affected by various pharmaceutical parameters, or various pharmaceutical or biochemical parameters, combining both. Okay, so let's first discuss with those parameters which are associated with the drug, that we call physiochemical parameters. It is drug solubility, permeability of the drug into gastrointestinal fluid across the GIT tract, high first pass metabolism, fasting or feeded state, pH of gastrointestinal tract, enzymatic degradation of drug, and variation is gastrointestinal mobility. So that is called peristaltic movement actually. These drawbacks of conventional oral dosage form can be overcome by designing a drug delivery system which can easily release appropriate amount of drug at a control rate to the desired site and reproducible result can be obtained. So, controlled drug delivery system release the medicament in a uniform manner to the site of absorption and also help to maintain the minimum effective plasma concentration for longer period of time. However, sometimes oral controlled drug delivery system can also get affected by various physiological factors like again the gastric intestinal mobility, pH, pathological conditions and fasting and feeded state. In uh, in that case, uh, if we look at the definition of controlled drug delivery system, keep in mind an ideal controlled drug delivery system should have two components. One, this is a spatial and temporal release pattern. A spatial release means a controlled release pattern over the period of time, and temporal, it means a particular in a particular area. So this is the situation for an ideal ideal controlled drug delivery system. In the case of oral, majority of area what we are achieving, or majority of characteristic that we are reaching is more about the temporal release, not the spatial release. Spatial release we achieve only by delivering through the IV route. Now there are different kind of oral drug delivery system, and among oral control drug delivery system, among one is osmotic control oral drug delivery system. Osmotic control drug delivery system is basically based on osmosis, principle of osmosis. So what is osmosis? Osmosis is the main phenomena which have been exploited for the delivery of osmotically controlled drug delivery system. And osmotically drug controlled tablet can be administered either orally or can be implemented in those part of the body where osmosis of the body fluid inside the device is possible. So definitely you cannot put this kind of delivery system just under the skin because there is no sufficient fluid. But in the GIT tract, stomach, which have a sufficient amount of biological fluid, another part like a small intestinal region, and some any other part of the body where you have sufficient amount of excess of body fluid, this kind of drug delivery system can be implemented or delivered. So the osmotically controlled drug delivery system is a sustained release system that work on the principle of osmosis and utilize the difference in osmotic pressure for controlled delivery or release of drug by using osmogen. What is this osmogen? Osmogen is also called as osmotically active material. These are basically the salt or the sugar solution. When it is coming into the contact of biological fluid or water, it dissolve and increase the osmotic pressure within that system. Uh, so the basic components of osmotic controlled drug delivery systems comprises of a core containing the drug, a semi-permeable membrane, in short form we say SPM, and a osmotic active agent or osmotic agent. So what is happening when exposed to body fluid or biological fluid? Imbibition of water takes place by osmosis through the semi-permeable membrane, and then uh, consequently osmotic pressure build up inside, which leads to the expulsion of drug out of the osmotic pump. Osmotically active controlled drug delivery system, which is one of the part of which is one of the content or type of a controlled drug delivery system. Osmotically active controlled drug delivery system, as indicated here, is the implantable controlled drug delivery system, which was first capable of releasing the medicament at a fixed rate for at least a week, and it was first designed by Rose and Nilsson in 1955. Then later on it was first marketed or produced in the market by the company Alza Corporation, which is based in California, USA, and that is actually first developed this osmotically controlled delivery tablets. This tablet was actually implantable. Alza Corporation has developed two type of basically the osmotic pump. Ortho is the abbreviation, so full form of Ortho is osmotic control release oral delivery system, and Durso. Ortho and Durso is actually the kind of osmotically controlled drug delivery system marketed formulation. Presently, more than 10 or so technology-based products are now is in the market, and they are for different kind of disease. So their application in obviously in different diseases depend on different kind of product that they are carrying. Now let's come to the Dorso. Orso was like a tablet. So now Dorso is like a small matchstick size osmotic pump. So now you know the difference between Orso and Dorso. This is based on the size. The first Orso was from the Elza company, and Dorso, the first Dorso was an implant approved by US FDA, is Viadur, that contain leuprolide acetate that is indicated in prostate gland cancer. So let me just brief you about the leuprolide drug. This is a hormone analog, and it is actually, just a moment. So leuprolide is basically, it is used in advanced prostate gland cancer, and it is gonadotrophin releasing hormone agonist. So I hope this is enough for your understanding. So now let's see how Orso and Dorso is having difference at the level of their principle or their structure. In an Orso system, we have like orifice, copore. That pore size you can control. Okay, you can control in micro. Now the development has evolved. You can also control in nano, and this is the pore that helping in releasing of drug from that pore. So now this pore is created on a semi-permeable membrane. This semi-permeable membrane is permeable to the water or aqueous system, but it is impermeable to something what is already inside in the beginning. So, and then there is a basic core in the structure called osmotic core, and that contains drug. Osmotic core means it also contains some osmotic active substance that once it will absorb the water, it will create high osmotic pressure, and that help in pushing all the content including the drug from the orifice or from the pore to the outside, which is the systemic environment or the blood. So these osmotic core containing materials or osmotic active materials are salts or some sugars that you will see later on in the slides. Now coming to the dorso. In the dorso, it is like a cylindrical shape, and in that cylindrical shape there is an alternate layer of drug reservoir and osmotic core containing material or osmotic active material are not together. They are separated by the layer. So you can see drug reservoir layer we have, and then we have semi-permeable membrane again, and then osmotic active agents layer, and then again semi-permeable membrane. So you see now the difference in their structure. Is a list of some marketed oral osmotic active control drug delivery system. So this is just giving you an overview that this is not something that is only at the level of lab. This technology is now well expanded or translated into the clinical practice. So elementary, there are some different kinds that already I had explained you. So elementary pump, push-pull system, some generic drugs that are already there is in elementary pump is phenylpropanolamine and enalapril and deltiazem, chlorpheniramine brand. These are some of the brand name that they have. Actune and Efidec, some companies which are now marketing this. As you see, most of the product and that was from the Elza. Elza was, you already have seen in the previous slide that Elza was the first company who developed this osmotically active regulated controlled drug delivery system. Now some more important companies in the pipeline that are delivering the product like Novartis, Pfizer, UCB Pharma. So now coming to the list, push-pull system. This is another kind of osmotic active controlled drug delivery system, and it contains verapamil, phenylphenate, isarapridine, glipizide. So mostly if you look at this, they are drugs from the category of anti-anxiety in the case of insomniac case, or it is also used in arrhythmic disease, like you can see verapamil. This is used in arrhythmia. Now some other product, that is Vidya-mitra, Concreta and Chromogeric. These are, and the drug that they contain is leuprolide acetate, which is obviously, as you have learned before, it is used in prostate cancer, and the other one is CNS stimulant, and chronogeric is used in chronic pain. So far you learned about the basics of osmotic controlled drug delivery system. Now, what are the advantages this system is enlisted here? It provides sustained release, that is zero order kinetic of medicament for longer period of time, and therefore we don't need to have a frequent dosing of the dosage form. So the patient compliance is better, side effects is less, delayed as well as pulse release can be possible. Drug release is not affected by GIT, pH and mobility when the osmotic pump is orally administered. This is the advantage when you can deliver it by the oral route. Drug release rate from the osmotic pump can easily be predicted. So in vitro in vivo correlation is easy to maintain or possible to achieve. Both kind of drug like hydrophilic and hydrophobic can be delivered using osmotic controlled drug delivery system. In previous slides you learned about the basics, applications and market status of osmosis controlled drug delivery system. Now here we are going to learn about the principle involved in designing or principle involved behind the osmotic controlled drug delivery system. As the word indicates itself, it is osmotic control. So there must be a process of osmosis. So let's first start with understanding of osmosis, what osmosis is. Osmosis is a process resulting in the net movement of solvent molecule into the region of highest solute concentration through a semipermeable membrane. So I'm just going to give you an example that you have a beaker. For example, in the beaker you create two partitions with the help of a semipermeable membrane. One partition contains high amount of salt. Salt is one of the example of osmogen or osmotic active material, and one part contain just simply the water. What will happen, that solvent will start moving from low concentration of solute, there is no salt at all, toward the high concentration of solute to maintain the equilibrium. That process is called osmosis. So this process contains till the solute concentration become equal. As I told you, it is a process of reaching equilibrium. Okay, when the drug containing osmotic vesicle is placed in the aqueous environment, what along with the water molecule, this drug start also following toward the solute concentration. So water molecule reach to the core of this device, yes, dissolving hydrophilic drug and escaping out through the delivery orifice. So this is about a water soluble molecule which is hydrophilic. In case of hydrophobic drug, what will happen, the drug device is fabricated in such a way that water molecule are not directly contact of the drug. Therefore a flexible ball or movable separator is used. So there will be a wall, or there is a wall of a membrane which is flexible, which if you put a push with the pressure, it start moving. During that, the drug come out of the device in response to the pressure developed by the osmosis. This is a process used in case of fabricating of hydrophobic drug. So as you know that this drug delivery system is based on osmosis. So this this principle basically governed by the equation of osmotic pressure itself, and it was Van 't Hoff equation, which is most commonly used to describe the mathematical derivation or mathematical principle behind this drug delivery system. So osmotic pressure, according to this, is defined as the pressure which need to be applied on the solution side to stop the osmosis flow of the solvent. So according to the equation, pi is equal to theta c RT, capital RT. Here the pi is actually osmotic pressure, and this theta is osmotic coefficient of the solution, and C is the molar concentration obviously of the solute, and R is the universal gas constant, and T is the absolute temperature which is presented in Kelvin. Let's come to the osmotic flow of water, because this is very important indeed. How much water will flow inside the system, it ultimately will influence the rate of drug coming out from the system. So how, because this is the point, or this is the parameter that regulate the rate of drug release. Okay, so how we control it or how we calculate it. So this is actually the extension of the previous equation. Osmotic flow of the water inside is governed by the equation dm upon dt in terms of mass. Okay, this is in terms of mass rate. A theta delta H, delta pi upon H. So dm upon dt is the flow of water across the SPM, semi-permeable membrane SPM is, okay. A and H, area of the thickness of SPM, it is again the area and thickness of semi-permeable membrane. Theta is osmotic permeability, and delta pi is osmotic pressure difference between the solute and the solvent. So in the previous slides, now you know the basic principle or mathematical principle behind osmotic control system. You learned that it is the osmotic pressure that governs whole system. Accordingly, now you know already the rate of inflow of water inside, which is directly proportional or it is in directly control of the pressure difference. Come to the basic components which is in osmotic control drug delivery system in terms of the design. Obviously, you are using this system to deliver the drug, so one component will be a drug, another component is semi-permeable membrane. Then you must need a osmotic active agent, that is also called as a term as osmogen. Then there is a layer called wicking agent, or a substance called wicking agent. Sometimes there is not always how much pressure and how much chemical you put it there, it not always leads to complete release of drug from the system. So to improve it, to increase it, we need something called flux regulator. Flux is nothing, flux is a parameter that shows the permeability of something against a layer. Pore forming agent, plasticizer, and coating solvent. In later slides we will learn about each individuals. Okay, so in this slide we are going to discuss what kind of drug that can be delivered using this osmotic control drug delivery system. The drug should have following property, like it should have a short elimination half-life, varied ranges from 2 to 6 hours. If the half-life is very less, it means within a few hours, then it will reach below minimum effective concentration, thus require repetitive dosing affecting the patient compliance. Also, the other problem with the short half-life that you need to intake more and more from drug within the formulation, so it will be difficult to manufacture. The other part from that is the compliance issue. Repetitive dosing administration may also produce some serious adverse effects. Another thing is that osmotically active controlled drug delivery system provide a solution to these above mentioned problem because of its sustained release characteristic. Hence, the drug with short elimination half-life are preferred. Another category of drug that is good for this is a highly potent drug. Drug with a small dose are preferred because osmotic device with such large dose is difficult to fabricate. Another requirement in this category is prolonged treatment. Drug which are used to be used for longer duration are preferred to avoid repetitive dose regimen. For example, diltiazem hydrochloride, metoprolol, nifedipine, glipizide, and methyl penidate. These are the drugs that really having moderately higher short half-life. You have seen in the list of content that drug, which is main important thing, and drug varied, but there are some parameters or some criteria. On the basis of that, you can select a drug and you can only make osmotic control system for that particular drug. That you will learn later. First of all, here the second most important part or important component of this system is semi-permeable membrane. In short form, we say SPM. What are the criteria, what are the characteristics of such membrane is? This kind of membrane must be inert, obviously, and biocompatible. So biocompatibility means it should not influence or should not create any sort of toxicity on immune responses in the body. It should not have soiling property, okay, and it should not be very rigid or very brittle. Be in a position, it should be strong enough to tolerate the pressure that is created by osmogen, okay, in the system, okay, and it should be selectively permeable to the solvent or biological fluid, or impermeable to other various large molecule or large substances. Obviously, it should be impermeable to solute, because you know that if it is permeable to solute also, then when the water will come, solute will also go out, and ultimately the reason where you want to create the osmotic pressure, you cannot. Okay, so that's why it should be impermeable to solute. It must only be permeable to the drug, so its selection is important according to the characteristic of drug. So here are some of the polymers that are used on the basis of parameter that what I had enlisted. Cellulose derivatives or cellulose ester derivatives. Some of them are cellulose acetate butyrate, and cellulose triacid. You can see that these are the most common polymer which is used in such drug delivery design. Next is ethyl cellulose and Udragit. Udragit is a marketed name, okay, and it is also a sort of cellulose derivative. Now, next important component. So you already learned drug. I will discuss with you later. You learned about membrane. Now you come to the osmogens or osmotic active agents. This is the material which is the core or heart of your osmotic control drug delivery system after membrane. Okay, so this is the material which is responsible for all what you want. Osmotic agents help in maintaining optimum osmotic pressure inside the system. So they are acting as a driving force for the water uptake, and drug uniformity is maintained in the hydrated formulation. So after absorbing the water, when the water coming inside, they got solubilized. After getting solubilized, they created osmotic pressure, and this osmotic pressure helping in pushing out the drug which is solubilized in aqueous phase. So these agents are, as I told you, every salt can be osmogens: sodium chloride, potassium chloride, sulfate of sodium, potassium and lithium. Some hydrophilic polymer that can absorb, okay, the what that can be dissolved in the state of water. Dissolve state is very important, okay, so be careful. Glucose, sorbitol and sucrose is an example. So they are basically the sugar molecules. This is the list that's showing you that what kind of material or osmogens you should use. Okay, because here is the list of the pressure exerted by the particular category of osmogens. But look, this osmotic pressure developed, what it is there, it is also a concentration dependent phenomena. Okay, so don't go directly to the data. Okay, this is just a data that is what is presenting you here, that okay, this can be achieved. Okay, but concentration is not here given here, and concentration is also important because if you increase the concentration, pressure that is listed here can be changed. But obviously if you want to increase the concentration, you want to use more material, and thus drug delivery size will change. So be careful for this. Most high pressure, if you want to create, lactose and fructose can give you the good pressure at a low concentration. Then afterward dextrose, these sugars are good. Sodium chloride and salts, other salts you can see below, salts are creating less pressure. So if you want to create more pressure using salt, you need to have more concentration. So it would be better if you just use sugar solutions or carbohydrate based materials that can create better pressure at low concentration. Okay, so the next important component of this osmotic control drug delivery system is wicking agents. Wicking agents is literally a piece of cord that convey liquid by capillary action. So it is actually a delivery cord, or delivery means biphasic, going and coming through, that based on the principle of capillary. The function of wicking agents are they help in activating the osmotic phenomena and carrying solvent molecules toward the core of osmotic device. Okay, the loose adhesion of solvent molecule take place due to the van der Waals force between the solvent molecule and wicking agents. Okay, so here is the example of some wicking agent that can be used, commonly used in osmotic active control drug delivery system. Bentonides, kaolin, alumina, titanium oxide, magnesium aluminum silicate, colloidal silica, niacinamide, sodium lauryl sulfate, SLS, that is SLS, most commonly abbreviation used for low molecular weight polymer polyvinyl pyrrolidone. Flux regulator, this is one of the important component or material used in the manufacturing of osmotic control drug delivery system. These are the material, okay, that used to fill in along with the permeation inside device So what is its purpose basically? Its purpose is to regulate the flux and increase the drug release through the orifice. They possess flexibility as well as the porosity. They enhance the porosity of the lamina of semi-permeable membrane. This is now come to the flux regulator. This is also very important material that is used in this system, and as I told in the beginning of the components slide, that the flux is basically used to improve the permeability, because cannot sometime the concentration of this osmogens is not optimum enough, or development of a optimum osmogen concentration is not good enough or critical to have a good permeability. So we are additionally adding this kind of regulator. Examples are polyalkyl glycol, for example polyethylene glycol, molecular weight ranging from 200 to 600, polyhydric some alcohol. Okay, so basically they are hydrophilic in nature because of OH group, and they enhance the solvent influx into the osmotic device, and therefore the osmotic pressure exerted inside is more, and you expect that more drug will release. So after all, the residual drug within the device can be reduced or minimized with the help of flux regulator. Some phthalates such as diethyl phthalate or dimethocrethyl phthalates are lipophilics, and they are also used. However, they are known to decrease the flux, so we should not use it. To come out from the doses, this type of drug delivery system, you must need a pore. Okay, and the size of pore, creation or technique of creating the pore is very important. There are different kind of agent that need to use for pore. Is called pore former. So used in the formation of pore. Multiplicate. Okay, pore former are water soluble in nature basically. Produce pore of different size depending on their concentration or whatever the nature. It can create the pore of from 10 angstrom to 100 micrometer. So when the osmotic device come in contact with the water, pore former get dissolved, leaving a microporous membrane, so form pores. So these kind of pore and their size are also a kind of rate limiting factor, a kind of parameter that can control the drug delivery. Some of the material used as a pore former are sorbitol, mannitol, lactose, glucose, fructose, sucrose. So they are basically similar to osmogens in some of the cases. Some are different, like polyvinyl pyrrolidone, some salts and amino acids. Now this is again another important component of such drug delivery devices. Coating solvent. A coating solvent is meant for dissolving polymer, which is supposed to be used during the fabrication of wall of osmotic device. This coating solvent either come from the organic solvent origin or may come from the inorganic solvent. However, whether it is from organic or inorganic one, it should be inert and should not have any unwanted reaction with the core material. So it must be inert with respect to the core material. So example of such coating material are water, it is aqueous base, methanol, ethanol, it is a kind of co-solvency nature with the water, purely alcoholic nature, isopropyl alcohol, some other organic solvents like acetone, ethyl acetate, carbon tetrachloride, cyclohexane, methylene chloride, these are commonly used as a coating solvent. Coating solvent. Coating solvents are one of the, basically when you are developing the system, this is something that is used from outside for the coating purpose, and they are often the mixture of a organic and water miscible or water kind or aqueous solvent. Okay, these are the ratio what is written here. These are some of from the literatures what we have find, and these are the optimized ratio what they achieve, or in the literature they achieve from certain experiments, but this can be vary, okay, depending on the case of formulation optimization. Okay, so these are some of the example that I am giving you here. Acetone water 90-10, so you can see that organic phase is more and aqueous phase is less. Also in the next case, acetone water, acetone is completely organic solvent, methanol is highly water miscible solvent, so normally it is used as a co-solvent. Next case, acetone, ethanol, 80-20 ratio, methylene chloride, methanol, it is again you can see that methylene chloride is higher in volume, Then the methanol. In some cases we use completely organic solvent, water miscible solvent, and some sort of water also. That is the last example indicating methylene chloride, methanol and water in 75:22 and 3. It is in this ratio. Now plasticizer, this is also very important component. This is provide the elasticity to the coating polymer and enhance permeability of the water. So plasticizer, it actually stop or it actually modulate or decrease the rigidity of the coating polymer. So plasticizer possesses a property which make the hard and brittle polymer softer, as I told you already, and form soft polymer can withstand mechanical pressure. They also mineraloid film forming property of polymer. 10 part of material which are supposed to form wall of the coat are usually mixed with 0.001 to 50% part of the plasticizers. Some of the example of plasticizer is phthalates, from the category of phthalates, triethylcitrine, polyethylene glycol, PEG 200 to 600, this is commonly used, ethylene glycol, monostearate, and diethyl tetrate or diacetyl. So let's summarize this whole chapter or whole module. uh, OCDDS, which is the abbreviated form of osmotic control drug delivery system, are novel drug delivery system working on the principle of osmosis for controlled delivery of or release of entrapped or encapsulated drug. It is indeed an excellent mean of delivering the drug in a sustained manner, and it is useful in case of chronic diseases. Osmotic delivered device, it can be given orally as well as it can also be implanted. Sorry, yeah. And another category of this is another osmotic device, majorly comprises of drug, semi-permeable membrane, and osmotic agent. The other component that form the part of osmotic device are wicking agent, plasticizer, flux regulator, pore formation, and coating solvent. Now, a numerous drug has been developed and marketed as oral as well as implantable osmotic control drug delivery system. Thank you so much.